On predicting cell cycle stage from 3D single nucleus strained images by Gangli.
Hello, everyone. Thank Anshu for the introduction. My name is Gangli. I'm a postdoc in the Bill Noble's lab. Today, I'm going to talk about our work to infer cell cycle stage from single-cell nucleus stain images. And today, my talk is going to consist of three parts. First, I will introduce the existing tools for the cell cycle prediction, and then I will introduce our method to predict cell cycle from the Fuji cell lines. And ultimately, we want to extend our work to other cell lines or mouse tissues. So first, I want to give you a brief review on how cell cycle, what is cell cycle. In the cell cycle, each cell grows and divides, and it has been known that it can be classified to different stages. For example, in G1, cell grows and prepare for the DNA replication, and in the S phase, DNA are replicated roughly twice, and in the G2, and cell grows and prepare for the mitosis stages, and M represent the cell division occurs. And there's also a small stage called G0, which represents when cells stop dividing. And there are many tools have been developed to estimate cell cycle stages, particularly from the single-cell transcriptomics data. Here I list 6 methods, but there are many more of them. However, a lot of those predictions from several methods don't agree with each other. And this is one way to infer cell cycle stages, but we are going to focus on how to infer cell cycle stages from image modality. One method can be used to infer is from the pCNA images, and research group from the UNC Chapel Hill, they have been able to use pCNA to assign cell cycle stages to study human cell cycle structures. They identify this manifold where you can see the cell cycle structures here, and including the G1 senescence cells on the tail of the left side. And they are able to classify cells to 3 categories, G0, G1, S, or G2. And this has been published in the Cell System 2 years ago. And we particularly focus on to infer cell cycle stages from the DNA-stained fluorescence microscopy images. And DAPI is one of the most easily to use and easy to get signals from the images. And this has been done to use in 2015 Nature Protocol paper. They have established a method using Gaussian mixture modeling to first get the integrated DAPI intensity after the 2D maximum intensity projection. And they provided a commercial software called FCS Express to modeling the 3 class cell cycle stages and estimate the cell cycle labels for each single cell images. Here, I summarize all the existing tools to predicting cell cycle stages from the DAPI images. However, those existing tools have several limitations. For example, most of the existing tools focus on the 2D maximum projections, which throw away a lot of the 3D cellular image information. And those image data sets are not directly available. You need to send emails to them. 2 of them did not reply to our request. And also, it would be interesting to see if we can extend these models to other cell lines or tissues. So to address those limitations, we developed a machine learning framework called Cell Cycle Net, which we release both our data and models, and it has already been put on the public GitHub repo. And we try to use 3D image information directly to improve the cell cycle inference. And we evaluate on both epifluorescence and confocal microscopes. So now I'm going to introduce our method. Before that, I want to show you how do we provide the ground truth label for the cell lines we collected. We specifically utilize the Fuji GUI system to provide the ground truth cell cycle labels. Here on the left, it's a cell imaging movie. As you can see, it's measuring the HCDT1 and HGEMLIN, 2 signals, to provide the ground truth cell cycle label. Initially, the cells are in red color, classified as G1, and then in the later stages, they show more green signal. They are classified as SG2M. So following the recent paper on Narutomo et al., we are able to classify the Fuji cell line data to 2 categories, G1 cells versus SG2 cells, using the Fuji system. And we collected 2 benchmarking image dataset with Fuji 2A systems. And here are the distribution of G1 SG2 dataset in the 2 most widely used microscopes, epifluorescence and confocal. And in both 2 datasets, we have a slightly more G1 cells. And next, I want to show you the data set we collected. Here is 2 morphology spaces. On the x-axis is the normalized integrated depth intensity, and on the y-axis is normalized volume. And as you can see, the green cells at SG2 cells, they are usually typically larger and brighter, and the purple cells here are the corresponding to G1 cells. We use Apricot, which adopts a submodular optimization algorithm to highlight the top 10 representative cells in this 2-dimensional morphology space. And here are those 10 representative cells we selected. So now I'm going to formally introduce our cell cycle night, which takes the 3D single-cell images directly to the input, and we fine-tuned a UNet model to predict the binary classification label, whether cells belong to G1 or SG2. And specifically, we utilize this UNet encoder, which to learn the 3D cellular information. And we fine-tuned an existing UNet, which have been trained on some cellular images. This have been published in 2011. And our baseline model is one of the previous tools, SVM model. This model takes 2 morphologic features, the 2D depth intensity and the total cell area as input, and train a spot vector machine model and output the cell cycle stages. And here I'm showing you one decision boundary using those 2 morphologic features as input to predict cell cycle stages. And one of the data set, epifluorescence data set, here I'm showing you the RC curves of our model, the blue curve, cell cycle net, compared against with red curve, SVM curve. As you can see from RC curve, our model significantly outperformed SVM model in terms of the classification performance. We achieved 0.95 error under the RC curve. And on the confocal data set, we achieve a similar performance improvement with our fine-tuned cell cycle net. And here is a test data set accuracy comparison. As you can see, both 2 data sets, our model significantly outperformed the support vector machine model. And next, we investigate whether our prediction errors are uniformly distributed across those 2 morphologic features. Here it's a same scatter plot, but colored by whether the cell cycle net is making correct prediction versus incorrect predictions. As we can tell, those black dots are uniformly distributed in this 2D space. And similarly, trends are observed in the SVM predictions, but we can see when we compare the predictions of cell cycle net versus SVM, here blue dots are the ones where are making correct predictions and SVM are making incorrect predictions. This improvement seems also be uniformly distributed in this 2D space. And one of the questions we have is whether we can incorporating 3D features to enhance cell cycle estimation purely from their SVM model. As this intermediate experiments, here the original SVM pipelines go from the 3D depth image. They do the 2D maximum projection and 2D segmentation to get those 2 morphologic features and train SVM to get cell cycle stages. So we proposed an intermediate experiments is to do 3D segmentation and also similarly extract 2 morphologic features, but it's 3D integrated depth intensity and the volume of the cell images, and train an SVM model to see how it performs. Interestingly, that we found that using 3D features with a purely SVM model significantly outperform using the 2D features for both data set. The blue curves represents the epifluorescence data set, red curves represent the confocal data set, and the solid lines are the model with the 3D morphologic features, dashed lines are the 2D morphologic features. And we also find ensembling those 4 morphologic features could further improve the performance. And here the blue bar is the SVM model with all the 4 morphologic features I mentioned before, and the rest 4 bars are just simply taking each morphologic feature to train SVM model. As you can see, the 3D integrated damping intensity is the most predictive single feature, and the cell area is less predictive compared with other features. And last but not least, we want to test the generalizability of our cell cycle net models. And we found that our cell cycle net model can benefit from learning across both datasets. Here, we are doing a training scheme where we are training on the confocal dataset or training on both datasets and test it on the epifluorescence dataset. And the dashed curve represents the model trained on both datasets. As we can see, for the SVM, the red curves, the model trained on the other dataset or both datasets doesn't seem to change too much. But for our dataset, if we can join training from both image datasets, the model got significant improvement in this generalizability task. Similar trend if we train on the epifluorescence dataset and apply to the confocal dataset. So after I establish our cell cycle net method, next I want to show how we plan to extend our current framework to other cell lines or tissues. Specifically, we want to extend this to the tissue environment. Here is mouse organ images. Here we have the DAPI signals. It's easy to extract the DAPI signal. However, it's hard to get the cell cycle dynamics. And you will be able to get different section, different morphology of cells. And the next question, whether the model can accommodate for those different morphology features from different kinds of cells. And we are able to use a Fuji2A cell cycle reporter model string for the ground truth. Here I'm showing you an example where we see in this mouse embryo images, there are certain regions are enriched for G2S cells. There are certain regions are enriched for the G1 cells. And we will generate those kind of images for the whole embryo scanning. And in summary, I think today I have shown you we collected those both epifluorescent and confocal image dataset with Fuji2A labels. And our cell cycle net achieved the state-of-art performance. And we generate Fuji2A mice and harvest embryos for imaging. Ultimately, we want to study the cell cycle dynamics for each cell type and tissue. And our code is available on GitHub, and our preprint is also available from this QR code. And with that, I want to thank my advisor, Bill Noble, and Brian Belleville's lab, Wal, Nico, Eva, Connor, and all the Noble Lab members. And I also want to let you know that our Noble Lab have 4 postdoc positions. If you are interested in the above-mentioned projects, you should talk to Bill Noble here in person.
Thank you.
Thank you.